Hey guys, Crazy King 58 here, and today we are doing a new series. Now this is going to be the AQA A-Level Biology spec. Now this is the Biological Molecules section, aka Unit 1. I've got a whole PowerPoint here that I've created based on Unit 1. This series is going to be made into a playlist by individual topics within this one unit. I'm going to be doing this for A-Level Chemistry and A-Level Statistics as well. So if you have any questions or any things you would like me to release first, put it in the comments. Any questions you have to ask, put it in the comments, and I'll be sure to answer them. Now, the first little subtopic we're going to be looking at within Unit 1 is carbohydrates. Okay, here's the little slide. So, carbohydrates, the main thing we're going to focus on here in this slide is monomers and polymers. So monomers are small units from which a larger molecule is made from. So like a monosaccharide would be a small unit of a polysaccharide. An amino acid would be a monomer of a polypeptide. And a nucleotide would be a monomer of a polynucleotide strand. Polymers are molecules made from a large number of monomers joined together. So like DNA is a polynucleotide strand, or it's made up of two polynucleotide strands. Polypeptides are made out of amino acids, and polysaccharides are made from a large number of monosaccharides. Monomers are joined together by condensation reactions. Now, a condensation reaction involves the formation of a chemical bond and the elimination of a water molecule. Now, that's important to remember. A hydrolysis reaction breaks the chemical bond between two monomers and involves the use of a water molecule. So if we look below, we've got sucrose plus water equals glucose and fructose. Now, this shows that sucrose is made up of glucose and fructose, meaning sucrose is a disaccharide. Now, the bond, which I am circling my cursor around right now, this bond here is called a glycosidic bond, and that is formed between the condensation between two monosaccharides, or between the monomers, glycosidic bonds form. Now, this water molecule is going to break that bond and put it back into its monomers. That's pretty much how a hydrolysis reaction works. Now, a condensation reaction is just the opposite. The OH group and the H group are going to form a water molecule, which is hence why it's plus water molecule, and it's going to make a disaccharide. Okay, now that's the basics. Now we're going to go on to the actual carbohydrates. So carbohydrates, now you see in brackets C, H and O because the element that they are formed out of are carbon, hydrogen and oxygen. Now a monosaccharide is a singular unit, so it's the monomer of a larger carbohydrate. The monosaccharides, which you need to remember, are glucose, galactose and fructose. A disaccharide is formed when two monosaccharides undergo a condensation reaction to form a glycosidic bond between the two monosaccharides. Hence the name di, because there's two of them. Now you need to remember the equations, or kind of the equations, to make the disaccharides. So glucose plus glucose gives you maltose. Glucose plus fructose equals sucrose. That was the example in the previous slide. Glucose plus galactose will give you lactose. Now maltose, sucrose and lactose are all disaccharides. Now, you should see two isomers of glucose on that. So you've got alpha and beta glucose. Alpha glucose has the OH group on the bottom and the H group at the top, whereas beta glucose has the OH group at the top and H at the bottom. That is important to remember. Polysaccharides are formed by the condensation reaction of many monosaccharides, so like the polysaccharides are starch, glycogen and cellulose. We'll look at those in a minute. Glycogen and starch are both formed by the condensation of alpha glucose, which is why it plays a role in energy storage, and cellulose is formed by the condensation of beta glucose, hence its role in mechanical support, like in cell walls. Okay, so now we're going to move on to the polysaccharide of starch. Starch is found in plants in the form of small grains. It is made up of alpha glucose, which are linked together by glycosidic bonds formed from a condensation reaction. Now starch can either occur in a branched chain, which is called amylopectin, or an unbranched chain, which is called amylose. Amylose is a tightly wound coil, so it's wound into a tight coil, which makes it very compact. Now this means a lot of it can be stored in a small amount of space. Starch's main role is in energy storage, and its structure is especially suited to this function because it is insoluble, so it doesn't affect the water potential, so water isn't drawn into the cells by osmosis. Because it's insoluble and large, it does not simply diffuse out of the cell. There's going to be more information on that on the transport PowerPoint in Unit 2, transport across membranes. Insoluble and large, so they won't go across the phospholipid bilayer simply. It's compact, so a lot of it can be stored in a small place as previously mentioned, and when it's hydrolyzed, so when water is used to break the chemical bond, it forms alpha glucose monomers, which are easily transported and readily used in respiration. Amylopectin has many ends, as is branched, so it can be acted on by multiple enzymes at the same time or simultaneously, meaning alpha glucose molecules are released rapidly, which provides energy. So starch is never found in animal cells, but glycogen is. Now glycogen is the next polysaccharide and is sometimes referred to as animal starch. So glycogen is found in animal and bacterial cells, but not in plant cells. This is the main carbohydrate storage in animals, and again sometimes called animal starch. It's stored as granules and is found in the muscles and the liver. Glycogen is suited to its energy storage role as it is insoluble as well, meaning it does not draw water into the cells by osmosis, just like starch. It's compact, so again a lot of it can be stored in a small space, and it's highly branched, so enzymes can simultaneously act on it, resulting in a rapid release of alpha glucose monomers, which are used in aerobic respiration. Now this is important to animals as they have higher metabolic rates than plants, as plants are less active than animals, hence why plants have starch and not glycogen. The next polysaccharide we're going to be looking at is cellulose. Now this is different from glycogen and starch, as it's the polysaccharide that's made out of beta glucose rather than alpha glucose, which is why it has different structural and functional features. Because it's made out of beta glucose, it forms straight chains, or straight and unbranched chains. We can see these chains like up here. These are cellulose chains. We can see this is beta glucose here. They're straight and unbranched, so they run parallel to each other, and because of this, allows cross linkages, so you can see these little dotted lines between the parallel beta glucose chains, or cellulose chains. Now these dotted lines are called, or cross linkages, are hydrogen bonds. Now hydrogen bonds alone are very weak, however the sheer number of the weak hydrogen bonds further strengthens the cellulose significantly, which also makes it a good role in mechanical strength. So this makes the cellulose good for support roles, like I just said, in the cell wall of plants. The cellulose molecules are grouped together to form microfibrils. So these are the cellulose molecules. Now they are grouped together to form a microfibril. And now a large number of microfibrils are grouped together to make a fiber or a macrofibril. Yeah, we've mentioned that. The structure of cellulose is suited to its purpose because they're made out of beta glucose monomers, so they form straight and unbranched chains. The cellulose chains run parallel to each other, which allow cross links to form, or the hydrogen bonds, and that further adds collective strength. These molecules are grouped together to form microfibrils, which in turn are grouped to form fibers and macrofibrils, which again provides further strength. So we can see here that goes there, and then that's in the cell wall. Okay, we're moving on to the tests. So this is how to test for reducing sugars, and we use the Benedict's reagent here, hence the name Benedict's test. So all monosaccharides and some disaccharides are reducing sugars, which means the sugar can donate electrons to another chemical or reduce it. A reduction is the gain of electrons, oxidation is the loss of electrons. The other chemical in this case is going to be the Benedict's reagent. So this test for the reducing sugars is known as the Benedict's test because we use the Benedict's reagent. Now, the Benedict's reagent is an alkaline solution of copper 2 sulfate. When a reducing sugar is heated with Benedict's, it forms an insoluble red precipitate of copper 1 oxide. So the colours differ, as you can see at the top. If it forms no colour change, or if there's no colour change visible, then there's none. If it has a green precipitate or a green colour, then there is a very low concentration. Yellow is a low concentration, orange is a moderate concentration, and red is a high concentration. So the test is as follows. So you add 2 cm³ of the food sample you want to test in a test tube, and you make sure it's in liquid form. If it's not already, just grind it up with water. Now you add an equal volume of Benedict's reagent. So if you added 2 cm³ of the food sample, you would add 2 cm³ of your Benedict's reagent. However, if you added water to grind it up, you would add equal volume of Benedict's, so equal volumes of the sample and Benedict's, no matter what. So after you've added the equal volume of Benedict's reagent, you would heat the mixture in a gently boiling water bath for 5 minutes. Now if the solution shows no colour change, there is no reducing sugar. If a green colour is present, then there has been a very low concentration. Yellow means low concentration, orange means medium concentration, and red means a high concentration. In other words, the more red it is, the higher the concentration of reducing sugars in your sample. The test for non-reducing sugars. So if your initial Benedict's test shows up with no colour change, then there is either no sugars present in your sample, or there are non-reducing sugars present in your sample. So how do you prove that there are non-reducing sugars present? So as previously mentioned, all monosaccharides are reducing sugars and some disaccharides are. So you add dilute hydrochloric acid, also known as muriatic acid, to your sample and heat it in a water bath that has been brought to a boil. Now this hydrolyzes the glycosidic bonds between the non-reducing sugar, resulting in its monomers. Now as it results in its monomers, that is the reducing sugar, hence would result in a colour change later on. So after you've done this, you would then neutralise your solution with sodium hydrogen carbonate, as Benedict's reagent does not work in an acidic environment. So after you've added the sodium hydrogen carbonate, you would use an indicator such as litmus paper to see if the solution has in fact been neutralised. If it has, you would carry out the Benedict's test as normal. You would add the Benedict's reagent to the sample and heat it in a water bath that has been boiled. You do this for 5 minutes. If a colour change occurs, like an orange-red precipitate, then a non-reducing sugar is present. The sugar has been hydrolyzed, so the monomers resulting from this would have an aldehyde or functional group, an aldehyde or ketone functional group that can donate its electrons to the copper 2 sulfate, allowing the precipitate to form. Now this is the end of the carbohydrate video. The next video is going to entail lipids. Now, if you have any questions based on this, just put it in the comments. If you don't, then don't. I would appreciate a subscription. If you want me to do more of these, or if you have any requests, just drop it in the comments and I'll do it. But yeah, that has been Crazy King 58, and I will see you in the next video.